The following content is provided under a Creative Commons license. Your support will help MIT OpenCourseware continue to offer high-quality educational resources for free. To make a donation or view additional materials from hundreds of MIT courses, visit MIT OpenCourseware at ocw.mit.edu.
I'm going to talk about a huge project. It's taken about 35 years so far. Lots of collaborators. I'll try to remember to mention them as I go along, but these are the key ones. Nat Alpert from the PET Lab at Mass General, Giorgio Ganis, Pierre Joulecoeur, and Bill Thompson. Let me start with 3 questions. So the first one is, what shape are Mickey Mouse's ears? Do you know the answer? Yeah.
They're round.
Excellent. All right. Which is darker green, spinach or lettuce? I'm going to pick on you again.
Always me.
Why not?
Spinach.
All right. And finally, in which hand does the Statue of Liberty hold the torch?
It started because there was no board in this country. I would guess right.
Excellent. So what I try to do here is get you to demonstrate yourself what I mean by imagery, at least from the phenomenological perspective. Does everybody have the sense sort of visualizing the objects when trying to answer these questions? If you looked at his eyes, they were moving to the side when he was answering them. If I ask you repeatedly, you won't do that anymore because you'll just memorize the answers. So what do I mean by visual mental image? It's a representation. So look at that word, representation, representation, that produce the experience of seeing in the absence of the usual sensory input. So it's a visual-like representation that comes from memory as opposed to online visual input. Talk has 3 parts. The first is the issue and initial studies. Second is neuroscientific data 1, focusing on the idea of depictive representations, and then we go to neuroscientific data 2, focusing on processing systems. So let's start with the first section, tell you what the debate is about. So this is from a paper by Zenon Polishin called What the Mind's Eye Tells the Mind's Brain. And it's really what kicked off the debate. Let me read a little bit from the abstract just so you can get it from his mouth. This paper presents a critique of contemporary research which uses the notion of a mental image as a theoretical construct to describe one form of memory representation. It is argued that an adequate characterization of, quote, what we know, unquote, requires that we posit abstract mental structures to which we do not have conscious access and which are essentially conceptual and propositional rather than sensory or pictorial in nature. Such representations are more accurately referred to as symbolic descriptions than as images in the usual sense. The issue is about the format of the representation, that is the form, the type of code, not about the content. So I could convey the information, the content, what I'm saying right now, in numerous different formats. I could write it out, I could say it in French, dots and dashes and Morse codes. Those are all different formats, different types of codes for the same content. The debate is focused on format. Is there more than one language of thought? Or is it all language-like propositional? It's focused on one particular alternative to propositional representations, depictive. So no one is denying that propositional representations are used. That's not the issue. The issue is sort of representational imperialism. Is it only propositional representations that are used in thought, or are there at least one other type of representations? I'm going to argue there's at least one other type, but I think there are many types. This is a thin edge of a wedge, getting the foot in the door. So let me just very briefly give you a sense of what I mean by depictive format. So here's some content. A ball is on a box, and here are 2 formats for representing that information. So on the left, I've got a propositional representation, which is descriptive. So the notation I've used is we make explicit the relation on the two arguments, ball and box. You can see there's an explicit relation. There are distinct arguments that are mentioned. There's a syntax. You can't just say on box. It's sort of dangling. Something's got to be on it. The truth value is true or false. It's abstract. Let's stop here for now. In contrast, here we have what I used to call quasi-pictorial, depictive representation of the same information, the same content. Here there's no distinct relation. On emerges from how this part of the drawing and that part of the drawing are juxtaposed. There are no distinct arguments. You can chop this thing up any way you want. It's a set of points that are juxtaposed. There's really no clear syntax, as Escher and other artists like that have pointed out. Truth value only when under a particular description. As Wittgenstein pointed out, a picture of a man walking uphill could be seen by a Martian as a man sliding downhill backwards. And it's concrete, it's not abstract. It's a specific example. Now we can run it in the other direction, where these kind of representations occur in a spatial medium. That is, they use space to represent space. So the way they work is each point on this representation corresponds to a point on the object. That's actually the planar projection of the object, such that the distances between the points in the representation, between that point and that point, that point and that point, correspond to the distances on the actual object. Okay, that's what I mean by depictive. So it occurs in spatial medium, and there's this kind of isomorphism. I don't have time to go into this in great detail. If anybody's interested, I can point out sources where it's been written up in great detail. So again, I want to reiterate that I'm not claiming that all representations used in imagery are depictive, only that some are. But that's the critical question, because it comes down to this issue of whether there's only a single language of thought. So the initial studies hinged on what I called studying the privileged properties of depictive representations. That is, what's distinctive, what's characteristic of depictive representations is not shared by descriptive, also called propositional, representations. The key idea here was it used space in a representational medium to represent space in the world. So the initial idea was, all right, let's show, using image scanning, that when you have the experience of a visual mental image, there's a representation in there, which is giving rise to the experience somehow, which uses space in the representational medium to preserve space in the world. So here was the concept. Try this, if you will. Visualize Snoopy the dog standing there. Now focus on his feet. Does Snoopy have pointed ears or round ears? What's the answer? Okay. Now try it where you focus on the center of his body first instead of his feet. Focus on the center of his body. Now tell me if his ears stick up or they flop down. Now focus on his head and tell me if his ears are rounded or pointed. Did anybody have the experience when they first started off focusing on his feet that they seemed to sort of scan up Snoopy to look at his ears? Did you get that sense? Okay, the idea was that if the representation uses space to preserve space, then the further you have to shift your attention, the longer it ought to take. So people often report if they start off looking at his feet, they got to shift further along to get to the ears than if they start off in the center of his body, and that's even less far than if they start on his head, which is right there. So here was an initial experiment to use response times as a way to try to externalize what's going on inside. So we're using response times as a kind of mental tape measure to try to use them to infer properties of the underlying representation. So this was a paper I published in 1973. It came out of my first year project as a graduate student at Stanford. It's called Scanning Visual Images, Some Structural Implications. So the idea was to use response times to try to get at the underlying structure of the representation. Had people memorize a set of drawings. They were all either horizontal, like these two examples, or vertical. The trick was there was something nameable on one end, like anchor, in the middle, like porthole, and the other end, like motor. They didn't know this when they were studying them. Later on, they were asked to close their eyes, visualize a particular object, like, say, speedboat, and then focus their attention on one side or the other, say the left side or the right side. And then they wait. And then they'd hear a property, like anchor or mast or motor, And they'd have to push one button as soon as they can see it in the image, and another button if they looked, quote unquote, their eyes are closed, it's all in the mental image, and they couldn't find it. Okay? And we measure the response times. They start off equally often focusing on the right, or the left, or the bottom, or the top. So here are the response times. Let's just focus on this for now. This is when it's on the side they focused on. Say the left side for the motorboat would be motor. This would be porthole for the motorboat, and this would be anchor. So the further they had to scan, the longer it took. And of course, equally often, the same item is here as here. So if they focus on the left, motor would be on the focus, and anchor would be on the far end, whereas if they focus on the right, it would be the other way around. So equally often, the same items are in both extremes. So it's not about the items. Here is where they're asked to keep the whole image in mind and not focus. So some of you, when you visualize Snoopy, may have kept the whole image in mind instead of just looking at his feet, in which case you wouldn't have to zoom. And that's what you show up here. It really is a function of shifting your attention. There was a verbal control group, which is irrelevant, it turned out later on, so I'm just going to forget that. So, did this experiment. I thought that kind of showed that when you have the experience of visualizing, there's something spatial in there. Wrong. Right after it was published, I got a call from Danny Bobrow, who worked at Xerox PARC, Palo Alto Research Center, who said it was a kind of cool experiment, but it didn't show what you thought it did. So I went up to visit him, and on his blackboard was something very much like this. What he said was, what you've shown is people have structural descriptions of shapes of objects, and that you can get them to activate a local part when you ask them to focus. So here's the motorboat, and what you've got is a set of propositions that are concatenated. So here's the relation, it's just a different notation. Here's the relation, bottom of. Here are the arguments, propeller and motor. So it's just like on ball box, different notation. And they're concatenated, so the nodes are arguments and the links are the relations, and it's describing the structure of that object. And what he claimed is when I asked them to focus on motor, they'd activate this local part, and then when they were asked about porthole, they had to go through 3 links, iterate through this structure to get to porthole. When they were asked about anchor, they'd have to go through 4 links, and that's why it took longer. This is a fancy way of saying that I was a first-year graduate student when I did that experiment, and it was a bad experiment because there was a confound. The further they scanned, the more things they went over. That's the confound. Okay? Furthermore, it was just one dimension. So here's another experiment where it's 2-dimensional, and we got rid of the confound. This map is set up so there are 7 distinct locations. They're marked by these little X's, and they were set up so that each pair of distances between all possible 7 locations is distinct. So the participants would learn to draw this map, actually learn to draw where the X's are and label them, like that's hut, that's well, that's pond, and so forth, beach. They get very good at this. Later on, they close their eyes, the map's taken away, they close their eyes, they're asked to visualize the map and focus on a given location, like hut. And then they'd hear the name of another object, like it might be tree or it might be bench, which isn't here. And their job was to shift their attention, push one button if they can focus on the next object, another if they can't find it. They start equally often on each object and scan equally often to every other object over the course of the experiment. Measure the response times. Here are the times from the trials where they could find the second object. So this is the distance between where they initially focused and where they ended up. And this is the time. And as you can see, it's a rather systematic relationship. That is, the further they had to scan, the longer it took. So, again, I was young and somewhat naive and thought this kind of settled the matter. Well, it ended up opening up a whole new round of debates. One portion of the debates focused on experimenter expectancy effects. So do you all know about Clever Hans, the horse that could add? So there's this famous horse that you could give it 2 numbers and it would tap out the sum with its hoof. And it was amazing. People were claiming it was reading minds and so forth. Anyway, it turned out what was going on is that people would be watching its hoof until it got to the right answer, and then they look up, and the horse had figured out how to respond to the cue and stop tapping. Pretty smart horse. The claim here from Margaret Intons Peterson is that our subjects are essentially horses, and the experimenters were the ones who were looking up. That is to say, that the experimenters were cueing the subjects about what the expected results were. So what she did is she told one group of experimenters that she expected faster times when people scan maps that are in front of them perceptually than in imagery, and vice versa for the other half of the experimenters, and sure enough it mattered. The experimenters who expected faster times in one condition got their participants to produce faster times. Okay, why? Well, they talked more animatedly and things like that. There were cues they were giving off. But if you look at the results, it was the intercepts that were changing, not the slopes. So the slopes are the increase in time with distance, and they scan, which is what's telling you about the underlying structure. That's what's reflecting the spatial structure. But the overall times are getting faster or slower in general. But so that it wasn't really affecting the part that was critical. But nevertheless, we worried that maybe experimenter expectancy effects could be producing these results. So Pierre Joulecoeur and I did a series of experiments. So in one of them, we brought in an experiment and we showed them this variant of that map. And we said, see these four objects down here? Well, they group to form a single unit by the Gestalt law of similarity, because they're all dark, and also the Gestalt law of proximity. So you have two of these laws, which are very well established, making this into a single perceptual group, which is hard to break up, we told the experimenter. So we expect long response times to scan between these, because they're part of a single unit. We also expect long response times to scan long distances, and medium response times for medium distances. So we drew out the graph, we predicted a U-shaped function, we told the experimenter. So he goes off, comes back after 16 participants, it's beautifully linear. He says, what am I doing wrong? We said, oh no, sometimes you need to test more participants to get a really stable result. Go back and test more. Tested 16 more, it was even more stable, linear. So we explained to him that we in fact expected it to be linear all along, but we needed him to believe that we expected U-shaped. He got very upset, he was pre-med. This is when I was at Brandeis, this was done, and it was very clear he believed it. Pierre and I did a whole series of experiments. We had colored maps versus black and white. We told some experimenters we expected the subjects, the participants, to be faster with the colored maps because they're more realistic. Whereas other experimenters, we said they should be faster with the black and white ones because they're simpler. It made no difference on the slopes. This is all published. It made no difference on the slopes. So it's not that the subjects are horses. Task demands. Here's another set of reinterpretations of our original results. This one from Zen and Polition. Paying attention. The imagery debate, analog media versus tacit knowledge. So the claim was it's not about picking up cues from the experimenter. It's about the way the task is defined. He says that the very nature of the task has built into it demands about how you're supposed to respond. So if you ask them to scan, unconsciously, they have tacit knowledge they're able to tap into about what it means to scan, and they believe that it should imply longer times at greater distances, and they have the motoric ability to regulate their response times to produce what they think are the expected results, all unconsciously. Let me, just as a footnote, mention that we've done experiments, we never mentioned the word scan. In fact, that very first one with the speedboat, the word scan was never mentioned. But let that be, okay? You get the same results. Let that be. The general point is well taken. Maybe it is the case there's something built into the tasks. So here's an example of a scanning task where there's no mention of imagery at all, let alone scanning. This is with ETL Dror. We presented this annulus with little squares. On every trial, 3 of them were randomly filled black. The participant would study the annulus with its filled squares. Would push a button when they're ready, it would go off, and then for 50 milliseconds an arrow would appear somewhere in the space that it did in the center, like here, except that they're not simultaneous the way I'm showing them to you now. They're sequential. And the task was to decide whether the arrow would have pointed at a filled square the way it does here if they were simultaneous, or would have pointed at an empty square. And the trick was there were 3 distances. This is the furthest distance, could be here, medium, or could be close. And of course it could point up, down, left, right, anywhere in the center region. They didn't know where the arrow was going to appear. And what you see is the further the arrow is from the square, the longer it took, even though the task was built so it required imagery, required scanning, no instructions to use imagery, no instructions to scan. As a footnote, by the way, ETL heroically lived on an Air Force base over the summer. This was part of a larger project. We were testing pilots, fighter pilots, to see if they had better imagery abilities. And we had this basic idea that imagery is not one thing. It breaks down to lots of subcomponents. I'll say a little more about that later. And that pilots should be good at specific ones. In particular, there's something called mental rotation. So try this, if you will. Visualize the uppercase letter N like Nancy. Can you do that? Rotate it 90 degrees clockwise. Is it another letter? Yes or no? If so, what's the letter? What?
Z.
Yes, it's a Z. So the further you rotate, the longer it takes. This is Roger Shepard did this work, in fact, at Stanford when I was there. I had nothing to do with it at all. It turns out the pilots are 10 times faster than Harvard undergraduates in mental rotation. But look at scanning. Here are the Harvard undergraduates. Here are the pilots. They're generally faster, but the slopes are virtually identical. So of course the question is, they get better selectively at rotating because they're having to deal with three-dimensional aspects of flying a plane, or is that something that was selected? That was the next study. Funding stopped. We never did it. But just as an aside, same slopes. Okay, so this was the first part of the debate. It was based purely on behavioral data. It eventually ground down. There were counter-explanations, new experiments done, went back and forth. In psychology, my sense is the attention span for any debate is maybe 5 years. And I think it was implicit, explicit memory was the next one that followed this as the hot thing. But I felt like there really should be a fact of the matter. It should be the case that if this is a science, we should be able to figure out when you visualize something, when you have the experience of visualizing something, whether there is something depictive in there or not. So I kept at it and came across some facts about the brain, which I didn't know. When I was trained at Stanford in the early 1970s, we were told the brain was irrelevant for understanding cognition. No kidding. Linguistics, computer science, philosophy, those were all relevant, but the brain didn't matter. It was the wrong level of analysis. So it's sort of interesting that I ended up turning to the brain, in particular looking at visual cortex. And the reason for that is I stumbled, literally by accident, on this drawing. I picked up an issue of Science magazine, opened it, and saw this. No kidding. This is from Roger Tutel, who's now at the Mass General, when he was a graduate student at Berkeley, in Devalue's lab. They trained monkeys to look at this pattern, staring at the center. These would be flashing lights all along here. The monkeys then injected with 2-deoxyglucose, which is a radioactively tagged kind of sugar substance. The more neurons are working, the more oxygen and nutrients are taken up, including, in this case, the tagged sugar. And this particular isotope gets lodged in the neurons, doesn't get broken down very quickly. So after the monkey had stared at this thing for about 10 minutes, it was sacrificed, and its brain was removed. And what you see here is the first visual area in the cortex, the first area that gets input from the eyes. And these dark lines show you where neurons took up a lot of the radioactively tagged tracer. And you can see that the right part of this pattern is physically laid out on cortex. This is the left cortex, so the right visual field goes to the left part of the brain and vice versa. So this is truly depictive in the way that I've defined it. If you have a hole in an area, which I don't recommend, but sometimes these happen, you'll get a scotoma, a blind spot, in the corresponding part of the visual field. If you have two holes in the cortex, the two scotomas will be positioned in space relatively far apart, depending on how far apart the two holes are. I mean, it's using space on cortex to represent space in the world, such that points on cortex, the distance between the points corresponds distance between the objects. Now, I have to be careful here because there's a magnification factor. The fovea, the middle part, is expanded. It's not linear. Okay, it's with respect to later areas in the brain, the way the connections work, it's being read off accurately. But if you look at this from the outside, the way we are right now, it's like a rubber sheet that's been stretched slightly. Nevertheless, it meets my definition of a depictive representation. So I found this astounding, because there I'd been talking all up to this time about there's no actual picture in the brain. I mean, who would look at it? There's no light. It would be uncomfortable. I mean, all that kind of stuff that Kosslyn had in his original article. You know, what the mind's eye tells the mind's brain. The homunculus problem. Meanwhile, so I was talking about it's more like in a computer when you have an array, where the array functions as if it's a coordinate space, even though the words in physical memory can be in different rooms as far as you're concerned. It's all vis-a-vis how a CPU reads in and organizes the bits of memory. So they function. It was all at a functional level. Function as if they're spatial. Well, it turns out in my very own brain, there were probably at least a dozen areas like this, where it's not just functional, it's also physically depictive. So this gives us a new way to approach the problem, which neatly sidesteps the problem of both demand characteristics and task demands. Because people don't know they have these areas in their brain, and they can't control what's going on in them voluntarily. So in humans, the corresponding areas in the back of the brain, it's called area 17 or V1. Here's the eye, so it would be like this from the side. So this is what I tell my undergraduates is a deep mohawk. It's cut right down the middle. So the eye would be there, it's the back of the brain. Now the way it's folded in complicated ways. The way the topography works is the fovea, the very center, high resolution part of the eye, projects very posterior along, this is the calcarine sulcus landmark, it's area 17 surrounding it, area 18 surrounds that. As something gets larger in the visual field, or parafoveal, extends more in the periphery, activation moves anterior, moves up. So this was shown in one of the very first studies of visual cortex with PET in St. Louis in the mid-80s. They had participants stare at a small pattern, I think it was actually red and green. It was oscillating right on the fovea, or sparing the fovea, another pattern that stimulates more parafoveally, or yet another one that spares the center and goes out more peripherally. Forget about this. And what they showed is, this is a photograph taken right from the journal, 1985, I think. There's an outline of brain. This is the resting baseline. And here's what was stimulated right on the fovea. It's the very back. And as it got larger, it moves up. As it got larger still, it moved further up. See, it's hard to see, but the blob is literally moving right up along the calcarine cortex, which shows that PET works. So we decided we'd take advantage of this finding and do the same thing with imagery, something very similar. So here's what we did for our first experiment. Close your eyes, please. Can you visualize the uppercase letter A? Tell me, does it have any curved lines, yes or no? Okay. Is there an enclosed region? Okay. Is there a vertical line on the left that goes straight up? How about a horizontal line? Any horizontal lines in there? Okay, so you can see it's a pretty good imagery task. We came up with 8 of these questions. In the scanner, we had subjects, participants, participate in 2 conditions. One where they visualize the letters as tiny as they could with their eyes closed. So they're laying there, it's PET now, so it's very quiet. Their eyes are closed, they're visualizing, they're hearing the names of letters. They visualize the corresponding letter one at a time, either as tiny as possible, and then they hear a probe, like curved line. They have to respond true or false. So we always have behavior so we can tell what they're doing. Or in another condition, they visualize it as large as they can without it seeming to overflow. The logic was, when it's tiny, there should be a part of cortex that's preserving a lot of spatial variation. It should be very active relative to this bigger version of the image. So right here, there's nothing, or there would be maybe one segment going through it. So we'd expect in the parts of cortex that are representing the very central regions, there should be more activation when it's a tiny image than a large image, whereas the parts of cortex that are representing the more parafoveal, peripheral parts, should be the other way around. If you're more activated with the large image because you've got something there than a small image where there's nothing there. So of course, we had half the participants start with the small image, half with the large image. They only responded to a given probe once in the course of the experiment. That was balanced. Everything's balanced. Here are the results. So 16 participants. This is when you subtract activation in the small versus large. Now I'm going to talk about the rest of this later. Let's just focus on the back of the head. So this will be the nose. This is the back of the head. Now, left and right are reversed. The reason for that is these technologies were developed in large part for radiologists, clinicians. And they're going to bedside, and they've got a scan they're holding, and the person's laying there. And what they don't want is the left side of what they see in the scan to correspond to the right side of what they're seeing in the bed. You have to mentally reverse it. So when you're laying down, if you see them from the bottom up, it's the right part of the head will be on the left. So by having the left and right reversed in the pictures, this lines up with what you see when someone's laying down, looking at them from the bottom up. That's why they're reversed, in case you ever wondered. So you see here, very posterior area 17 is more activated by the small image than the large, whereas when you look at what's more activated by the large image than the small, it moves forward, and it's actually bigger activation. Here's the same data seen from the side. Here's small minus large, so the eye would be here. You see it's very posterior. This is the calcarine sulcus, and here is when it's large. Isn't that cool? So it worked out. So I was very happy with this. This was part of the first PET paper we published on this, published in the early 1990s. The trouble is, a couple troubles. One is there were only 2 points. So someone could argue, well, it's not really a continuum, it's a difficulty issue. Maybe that's what's going on. That's problem one. And a more serious problem is, around the same time we published this, 3 other groups published papers on mental imagery that involved neuroimaging. There was Denis Lebion down at NIH, one of the very first fMRI studies. He got it to work. He got nice V1, area 17 activation. But then there were 2 studies in Europe, one in Karolinska in Sweden and one in Paris, that weren't finding area V1. So why not? So we looked very carefully at the papers and noticed that the 2 European studies used a resting baseline. They compared the activation during imagery to a resting baseline. And we thought, well, maybe during rest people are visualizing, maybe they're daydreaming. So you're comparing imagery to imagery, so no wonder there's nothing left over. So what we did is an experiment where there are 4 conditions. First, the first condition is, before they knew anything, they would be in the scanner and they'd hear the names of objects like guitar, and then they hear something like higher than wide, and they just alternated responses. Then they'd memorize, after they did that, a set of drawings, or I think 24 each set, and they were told, we're going to ask you to visualize these, so memorize them well. And then they were given the probes that we're going to be asking them after they memorized them. And then they were shown one of 3 squares. One square that was very small, subtended only 1/4 degree of visual angle. One that was medium size, 4 degrees of visual angle. One that was quite large, 16. This is from their point of view in the scanner. They'd be shown the square. And they were told, we want you to visualize the objects at this size. So if you hear guitar, we want you to visualize this drawing, the one that you memorized, the guitar, at this size, or whatever the size was for that condition. So far, so good? So we, of course, counterbalanced the order of these 3 conditions and which objects occurred in each. And the trick is that we'd ask them, is something like this, the right side higher? So for this drawing, the right side was not higher. The left side is higher in the picture plane, whereas for the anchor, the right side is higher. So to get it right, they had to be using an image of that particular drawing. And of course, we explained what the probes meant and so forth. So there were 4 conditions that involved the same tape. Okay, so we counterbalanced that so that the same identical physical stimuli occurred equally often in the 4 conditions: a listening baseline, and then the 3 imagery conditions, which differed only in the size of the image. We also had a resting baseline, which was either the beginning or the end, where we actually went out and bought the same blindfold that the Karolinska group used, and we used the identical instructions, which were actually to visualize blackness, believe it or not. That's what they asked them to do. So there were 5 conditions altogether. 3 that were imagery, 1 that was a listening baseline that used the same stimuli, and 1 that was just rest. So here's what we find when we compare to the listening baseline. So it's identical stimuli to the 3 imagery conditions: small, medium, and large. So you see the small images activate the very back of the brain, medium it moves forward, and the large, it actually is a bigger swath. It's all in the right hemisphere, by the way, which is kind of interesting. Very little in the left hemisphere at all. Now look at the identical data, imagery data, compared to the resting baseline. You notice something? There's nothing there. So when you look at, I was kind of puzzled by why other groups weren't getting this V1 activation. There's a whole system involved in imagery. I'm just focusing now on the depictive representation part, which is the very back of the brain where we have these topographically mapped areas. So let's just look at this for the moment. So here is the average regional cerebral blood flow during the listening baseline. One of the nice things about PET is you get real numbers out. We normalize the brain to 50 milligrams per milliliter per minute per 100 grams of flow, but relative to that average for the whole brain of 50, you can see during the listening baseline there's relatively little activation. And now look at during the resting baseline. Massive activation in area 17. This was well known, by the way. In fact, one of the classic works in this was out of the Karolinska. So we knew during rest there was a lot of activation in the back of the brain, and you're subtracting this as the baseline. That's why you're removing all the imagery baseline, all the imagery activation. So you could say, well, wait a minute. Maybe since this is relatively small, by subtracting this, you're just getting noise left over during imagery, and that's why it looks like you have something. Well, that's one of the reasons it's so important that we had 3 sizes. So it's not just that we activate it, we get systematic relationship between the size of the image and where the activation is. I actually decided to collaborate with them. So I moved my 3 little kids and very cooperative and wonderful wife to Paris for a year to work with the French group to try to figure out why they weren't getting it and we were. So we designed an experiment with the idea I had was it had to do with resolution, spatial resolution, and their tasks didn't require enough fine-grained decision making. So we designed a task where they had to compare relative heights along sets of geometric forms. It didn't work. It didn't work. And I had a major role in designing this. So I was kind of depressed. So the question was, well, are these topographically organized areas functional imagery? Maybe it was just some kind of a spin-off. Maybe it was epiphenomenal, like the heat from a light bulb when you read. The heat plays no role in the reading process. It's epiphenomenal. And that Kosslyn was claiming that the experience of visualizing is epiphenomenal. It doesn't reflect the underlying representation. Maybe he was right. So we decided to see if, in fact, this activation in V1 is playing a functional role before going much further. Here's a task. Participants memorize this pattern, set of patterns. There's 4 sets of stripes. They don't realize it, but they vary in 4 ways in terms of the width, the separation between them, the length, and the orientation. They're told this is quadrant A, this is B, this is C, this is D. The task was later on, well actually there's 2 conditions. One where they've got it in front of them, like you do now. They might hear A, B, width. So AD rather, AD, which name this quadrant and that one width. And the question is, are the stripes in the second named quadrant larger on the dimension? So width, yes, these are wider than those. Tilt, no, these will be tilted further from the vertical than those. Do you all understand what the task is? So we first did this with imagery. During PET, this is the brain seen from the back. This is right up the barrel of calcarein. You can see it's very nicely activated, V1. So it's a good imagery task. We then did it with TMS, transcranial magnetic stimulation. So the way that works is you put a current through a coil, creates a magnetic field. It's quite a strong magnetic field for a brief period of time, and this is a technical term, discombobulates the neurons directly under it, about a cubic centimeter. So you can put it right over motor cortex, motor strip, and activate individual fingers. I've had this done. It's really wild. It's really creepy. Or you can move it, so we did this, we got to calibrate the strength, then we moved it to the back of the head and aimed it right up the calcarein cortex in one condition, or so it missed visual cortex altogether in the so-called sham condition. So I was a subject in this. You're not going to see my data because I knew what was going on. But I thought the conditions were reversed because it's pressed very hard in the sham condition. The trick is you do it for about 10 minutes, once a second, and after a while, the neurons, every time they get this burst of magnetic field coming in there, they get sort of, I'm going to anthropomorphize my neurons, sick of firing. They start to adapt, and they don't get so responsive. So Alvaro Pascaleone, who is my collaborator in this, talks about a transient lesion. That, to me, that's kind of scary. It's not really a lesion. It's just kind of, they get kind of, I don't know, limp or something, less responsive. So after this is done, you have some time, you can do some testing with a brain area is less responsive. So that's what we did with that task I just showed you a second ago, where there's two conditions. They have it in front of them. That's perceptual. They were being asked to compare these different pairs. Or they've memorized it and their eyes are closed, and they're doing it from imagery. Each of these lines are data from a different person. This is sham, where it's missing V1. This is real. That's their vocabulary in that field, where it's actually going into V1. So this is during perception. So what you see is for every subject, and these lines are from different people, they're taking longer to respond if we had previously stimulated V1 than if we'd done the sham. So this just shows that V1 is important in perception. We knew that already. But if it didn't work, then there was no point in doing the imagery part, right? So this sort of validates that we're doing it right. Here is the imagery component, and what you see is, again, for every person, I mean, it takes longer in general. Imagery is a little harder than perception. You have to generate the image and look at it. But for every one of them, they're taking longer if previously we'd stimulated V1 than if it were sham. If you actually average these together and look at the difference in time, it's almost identical. The effect size is really quite similar. So then you could say, well, wait a minute. We know that from Zecchi's work that some of the stimulation travels. So if you use TMS in a given part of the brain, it may affect other parts. So maybe, again, it's epiphenomenal from some other areas. So we use statistical technique to try to get rid of that idea. So with this task, their eyes are closed and they're visualizing uppercase letters, and we get the response times out. What we now did is regression analysis. So here are data from each person. There's 16 people. Each dot corresponds to a measurement from V1 of how much blood flow there was during that task. And so here's the amount of blood flow. The brain is set, so they're normalized for each person to be a mean of 50 milliliters per minute per 100 grams of flow. And here's the response times from that person, for the different people. And what you see is the 4 people who are slowest are the ones with the least blood flow. It kind of looks like a step function. I fit a line through this, but it really looks like a step function to me, which is kind of cool. What we also did, this is all published by the way, is looked at every other area that was activated and regressed the response times against those areas. In fact, we forced, in some of the analyses, we forced in initially the contributions of other areas and removed them, and still V1 significantly predicts the response times. So it's not just an artifact of other areas. Not just an artifact of other areas. So finally, what about all those other studies? There are now 60. This was published a while ago. When I did this analysis, we did it, Bill Thompson and I. There were 59 studies, fMRI, PET, RESPECT. We looked at them carefully. There were 21 variables that distinguished them. Things like whether their eyes were open or closed, whether it was in the dark or not, whether it was a resting baseline, whether it was a spatial task, like the Statue of Liberty one, left or right, or whether it was a shape-based task, 21 things. So we did a meta-analysis. We coded each study 0-1 in terms of whether V1 or V2 topographically mapped, depictive representation mapped areas in the back of the brain were activated or not, and then use logistic regression to figure out which variables were driving the results. And it turns out virtually everything is accounted for by 3 variables. That is whether the task required high-resolution details of shape, like what shape are an animal's ears, whether it was non-spatial. So it turns out if it's a spatial task, requires a spatial relation judgment, you get a lot of parietal lobe activation, not V1 or V2. So that was kind of cool. So we coded this as non-spatial because otherwise it would be negative. We just didn't want that. And then technique. So if you have a 3 or 4T fMRI, you're going to get it. If you have a PET, you may get it. If you have SPECT, you probably won't, just in terms of power. Finally, last bit. So going back to the paper that kicked this off, What the Mind's Eye Tells the Mind's Brain, a critique of mental imagery. What he's driving at with this title is this apparent paradox of who looks at it, the homunculus. You need a little man in the head to be looking at pictures. That just doesn't make sense. So we wanted to say goodbye homunculus. The working hypothesis was there are shared systems in visual perception when you're registering what's out there via your eyes. And imagery when it's being constructed from information previously stored in memory. I've already given you some evidence of that, just looking at V1. But in fact, there are many areas. There are at least 32 distinct areas in the monkey brain that are involved in vision. So here's an unfolded macaque cortex. Seen from the side, the eye would be here. This is V1. The macaque, it's on the lateral surface. You saw this already. This is V1. Many other areas involved in vision. We know a lot about them. This is an old slide, but it makes the point I want to make. Here's V1 again. So the eye would be feeding. So this is bottom up, going deeper into the system. Notice that there are no arrowheads. The reason for that is virtually every area that gets an input, an afferent from another area, also receives feedback from it, efferents. Huge amount of information flowing backwards in the visual system. So old idea goes back prior to William James, actually. In fact, it's in his Principles of Psychology in the 1890s, was the information flows backwards during imagery. So we know in the temporal lobe, in the monkey, there are representations that store visual information in memory, but they're not depictive. They look like they use a population code. They're columns that code rather abstract visual characteristics, and it's combinations of activation within these columns that are storing the information. You could think of it as a kind of compressed code. Sometimes I use the metaphor, it's like you have a DVD, and if you want to reconstruct what the information is there, you need to unpack it and run it backwards, reconstructing the spatial information. Okay, there are signals that go backwards. It's more complicated than I'm letting on, but this is the basic idea. So the notion was the same system that's there in perception can be used also in imagery. I don't think it evolved, by the way, for imagery. I think it's like Stephen Jay Gould's idea of excitation. So you got a nose. The nose evolved presumably to warm air and to direct sense and so forth. But once you've got the nose, you can then use it to hold up your glasses. So these backward connections are probably there for cooperative computation. So later areas can send information to earlier ones to sort of help them. The system's working in cascade. It's like waterfalls. Everything's running at once. It's not like, this works, and then when it's done, it sends it to that, and then it's not lockstep. Everything's running at once. So in perception, it's useful to have information flowing backwards to help the earlier areas based on what's known, to help them converge. Once you've got those connections, the idea is you can use them, even in the absence of the immediate sensory input, to reconstruct information from memory. So we did an experiment where we again used this task, which I told you about before. We had them either now memorize these drawings or see them, but we made them very dim. So the idea is we wanted to get top-down processing involved during perception. So now, in the imagery case, they hear the name, and they have to visualize it, eyes closed, and they make a judgment. In perception, they hear the name and see it, and make the judgment based on what they saw, not having seen them before. And here's what you find. This is fMRI with Giorgio Ganis and some others. So here's the eye. The top row is right behind the forehead. And as we go down the rows, going progressively more posterior. So this is the front of the brain. So what you have on the left is perception, what's activated. In the middle is imagery, the eyes are closed. On the right is what you get when you subtract the two. Look at this. Isn't that incredible? There is no difference between their eyes being closed in imagery and their eyes open in perception in the front of the brain when they're doing this task. Now look at the back of the brain. So now it's the back third, posterior third. So this is the very back of the head, and this starts about two-thirds of the way back. So now if you start looking on the right here, you'll see there are differences. As you get further back, you're getting more activation in perception, where it's being driven by the world, than in imagery, where it's being driven by memory. Okay, it's about a 92% overlap. So virtually same system being used in imagery and perception, not identical though. There are some areas that are more activated in imagery than in perception, and vice versa. Of the 16 people, 12 of them showed good activation in V1, which is about what we find. About 20% of people don't show it so much. There are big individual differences on this. It's very consistent. Okay, let me summarize. The issue is format versus content. We're focusing on format. Is there more than one way to code information? I'm not disputing that we have linguistic-like representations. The issue is, do we have some other way to do it? What I've been arguing here is yes, we also have depictive representations, where space in a representation is being used to represent space in the world, such that points in the representation correspond to points in the world, and the distances among the points in the representation mirror the distances of the actual points on the object. There were 3 parts. The first was initial studies where we used behavioral techniques. I just gave you one example. There were other sorts of experiments. I used scanning as a kind of mental tape measure to try to show that when you visualize, when you have the experience, there really is something in there that's using space. The logic was the further you have to scan, the longer it would take if there's something that has space in there. You saw we went back and forth with various counter-explanations. It really never was conclusive. So then we turned the brain, taking advantage of the fact that there are truly depictive representations in the brain in these areas that are called topographically organized, and try to show that when your eyes are closed and you visualize, these areas are not only activated, but properties of the image affect how they're activated. So a small mental image, one that seems to subtend a small visual angle, activates more posterior, just as a small picture would, or a small object, that is small visual angle, not size the actual object. So we showed that. I didn't show you, we also done it with vertical horizontal, where they visualize vertically versus horizontally, and you can actually see it shift along the meridian for most people, not everybody. We also asked whether these representations were functional, showing that if you use TMS to disrupt processing in V1, it does affect performance in at least one imagery task, the one we looked at, which activates that area. And we showed using statistical techniques that the activation of V1 is actually functional. It's not just an epiphenomenal, and some other areas are actually driving performance. Finally, we asked the question, well, how do these representations function embedded in a processing system? I mean, something's got to be using the representation, or it might as well not exist. It's like that old saw, if a tree falls in the forest and no one's there to hear it, is there a sound? What's the answer, by the way? If a tree falls in the forest and there's no one there to hear it, is there a sound?
The answer is yes.
The answer is no. Sound is a psychological phenomenon. There's a distinction between the psycho and the physics. That's why they call it psychophysics. That is, the physics are there, you're right. There's a propagation of compression waves in the air, but that's not sound. Sound's got to have a brain event corresponding to it. In fact, you can get sounds by stimulating neurons without even going through the ear. I'm sorry, that's a trick question. So the idea was that, the reason I'm bringing this up is that without some processes that operate on representations, they may as well not exist. That it's only in the context of processing system they actually even have any properties, because they're being interpreted in certain ways and having certain consequences. So we showed that, in fact, most of the visual system is involved in mental imagery as well. So is there a little man up there? Well, if there needs to be a little man during perception, then I guess we're going to have to be stuck with it. But if there doesn't need to be a little man during perception, then there doesn't need to be one in imagery either, because it's the same system. So I've been doing this work since I was a graduate student, which was 1970. How many people here were born in 1970? Before then? There's a few of us. It's been a long project. And sometimes when I give this talk to younger audiences, they kind of yawn and say, this is all obvious. I mean, why'd you go through all this trouble? Which I find somewhat amusing, but in some ways comforting. But I like to point out that as we did all this work, we got some new insights that weren't obvious along the way. Remember those 3 items that I used at the very beginning? What shape are Mickey Mouse's ears? That's a shape question. Which is darker green? That's a color question. In which hand does the Statue of Liberty hold the torch? Spatial question. Well, it turns out, although it's not evident introspection, shape, spatial, and color imagery rely on distinct neural systems, partially overlapping but distinct. Shape processing uses parts temporal lobe that are not involved in the other 2. I already mentioned that spatial imagery uses parts of the parietal lobe, and there's yet another part, the fusiform gyrus, that's involved in color imagery and not in the others. So one of the things we're doing in this project is what's being done in cognitive neuroscience more generally, which is to pull the meatloaf apart at its joints. That is, to take functions that seem intuitively to be unitary and undifferentiated intuitively and show that they're not. They're actually composed of systems, of distinct kinds of entities that are interacting. So that's been one product from this project. If anybody's interested, I'll end with a tiny plug. There's a book where we just summarized all this in great detail. This is with Bill Thompson and George Oganis, my main collaborators, The Case for Mental Imagery. I'm not advocating that you search inside, but if you're so interested, it's out there to be looked at. Thank you very much.